First, just a few words of thanks to all of you for coming. First of all, thank you very much. And for the wonderful speakers today, it's a great honor and pleasure to hear from all of you. I'm humbled to be named as part of this lectureship of this Jakob-Herz, who I've learned a lot about over this period of time, and I'm honored and humbled to be named as part of his legacy. And he's such a pioneer that I feel a part of this whole ceremony. It makes me very proud. I also am very proud of the university. This is a forward-thinking and thoughtful, daring university to move forward over these years on these important topics. The city of Erlangen and their courage and forthrightfulness in honoring what should be honored and continues to need to be honored is very important, and I'm proud to be here in the city at this time. I'm also particularly grateful, it's been mentioned several times, that I was able to have several students, fellows, early in their career spend time with me in the laboratory. Obviously starting with Jürgen Winkler as early as 1992, and then two of his young fellows that followed in his footsteps, Chi-Chong Lee and Beata Wiener, who more recently has returned to Erlangen. All of this makes me actually quite emotional and proud to be a part of that. What's wonderful too is even students of Beata and Jürgen and Chi-Chong have come to spend periods of up to a year in my lab, helping us, learning, and I feel quite attached to FAU, and I'm very proud to be here. Finally, but not last, I have to thank my wife for, I should tell you that it's not just, it's those of you that have worked in my lab, and there's actually several of you here, realize that she's not just my companion and my wife and my best friend, but she's also a scientific editor. So she's taught many people in my lab how to write scientifically and continues to edit our manuscripts. So I get her free work there. She works on the outside, but she also edits our manuscripts and grants. So for that, thank you very much. Now I'm going to talk to you about, I was asked to present some science, and I'm going to tell you, if I can remember what my password is. Since I do work in memory, this should work. Close this off and start with this. No signal. Should be coming up soon. Okay. So I'm going to give you two topics. I'm not going to make too many assumptions about your background knowledge, and I won't go into too much depth, but I will give you a little bit of update on this concept of adult neurogenesis, in particular, something about what we know now about what it does. And I'm going to tell you a little bit about what we think its function is and why we have this process going on in our brain. And then I'm going to give you a little test. And that test is a way for you to get a sense of how your brain is functioning when these brain cells are active. This is all part of a sort of a new activity. I've got a feeling this is going to come up over again. First of all, when I came into this field, there was some resistance to the idea that the brain could make new neurons, and it's for two reasons. One is that if we're making new neurons all the time, how do we have any sense of self? How do we retain information if the neurons are turning over all the time? That was one of our major problems. And the others that were scientifically oriented, they knew what a neuron was and how complex it is with its processes and many connections, and how could a neuron divide? That just didn't make any sense. And the resolution to these two problems was that not all the brain cells in the brain are turning over, but they're only turning over in a few areas of the brain, very critical areas of the brain. And finally, in the adult, it's mainly the structure of the hippocampus that's involved in learning and memory, where these new neurons are being born over time. With regards to the dividing of complex neurons, a part of the discovery was that the adult brain retains stem cells. So there are, in fact, stem cells that exist in the adult brain, and these are the cells that are dividing and giving rise to new neurons throughout life. So there was the solution to the second problem of neurons undergoing cell division. One could understand this concern because, as Jürgen mentioned, we have on the average of 100 billion neurons and 100 trillion connections between these neurons. And these cells that are embedded in our brain are highly connected. So this structure where neurogenesis takes place is the hippocampus, and it's a structure that sits in our temporal lobes here and is crucial for the acquisition of new information and the transient storage of information. And when we recall information from the past, the information passes through from the cortex back into the structure, where we use this to determine whether or not the information that we're attending to at any one moment is a new piece of information or an old piece of information. So it's not just involved in the acquisition of information, but recall and recognition. And we must give credit to those that identified this, and some of you clinicians will remember, especially in the areas of psychiatry and neurology, this patient, and oftentimes we have to give credit to the patients. He was a epileptic patient who had both sides of his temporal lobes removed by Penfield, and as a result of this bilateral loss of hippocampus, he showed this loss of episodic memory, where he had every day was a new day for him. His past memories were fully intact, but his ability to acquire any new memories at all were vanquished. So over the years, we and my colleagues, now many other people, have demonstrated that this process of adult neurogenesis takes place in all species, all mammalian species that we've seen. And all mammalian species have this structure called the hippocampus in pretty much the same location. And not only that, the structure itself looks quite similar. So it has this same pattern. This is actually in the human hippocampus, but the molecules and genes that are involved in making these new cells are preserved across the entire phylogenetic phyla. This is just one of a picture showing this process, and I'll just describe this to you. This is a virus, innocuous virus that has a color green, which can infect only dividing cells, was injected into the hippocampus of an adult mouse, and it only survives for about 2.5 hours, and then the virus vanishes. So it can infect just a small number of cells over that period of time, and now this is 2 months later. So all of these green cells are newly born neurons that were born within a very short window of time, but they persist for the lifetime of the animal. And this is we found to be true in all species so far. So not only are they making new neurons, but they're contributing to the circuitry of the brain. I have to tell you a little bit more about this process of neurogenesis, but also the process of memory. This is the cartoon of the structure that we've looked at, and this is the cortex coming in. So memories are made by virtue of the sensory experience, the auditory experience, the olfactory experience that you have at any one moment in time. And what happens, for example, as you're listening to me, you're watching me, you're hearing me, you're having some reflection on what I'm saying and trying to make sense out of it, and also you're translating it into another language. But you're sitting next to people that are either familiar to you in terms of their odors or other features of them. All that information is coming in to this structure on the side called the entorhinal cortex, and it flushes through the hippocampus, where the different sensory processes are integrated into a single memory, and then it goes out into the cortex. Now, when you, in a month from now, when you try to recall this event, you won't see it as separate events, as just hearing my voice or just seeing my face. If you remembered it all, and let's hope you remember some part of it, it'll come back to you as an aggregated memory, as a single memory, which integrates these different components of the process. And it's really this structure of the hippocampus that aggregates these different elements of an event into the form of a memory. And what I'll tell you is that, and what we've learned is that these newly born cells that are right in here that are being born into the structure are important for being able to make discriminations between what you're seeing right now as being new and something that is slightly different. If there's a major difference between the two, you really don't need these new neurons. They're only important for making distinctions, subtle distinctions between events. I'll come back to that briefly and give you a test on this. So, in summary of 20 years of work, we know a lot about where these cells are born and the surrounding environment that they have and how long it takes them to mature out into full neurons and the biology of their role during this process. I'm not going to go into that in any more depth. But what I will say is that what I think is quite remarkable is it doesn't just happen on a regular basis, but the process is influenced by what we do. So the number of neurons that come into our brain and integrate into their brain and the role that they play in our past memories is affected by what we do. So if we live in an enriched environment where we're learning new things all the time, more cells will be integrated and more cells will survive and have much richer integration. If we're physically active, moving around where we're increasing blood circulation, the total number of cells within the hippocampus that are being born increases. The amount of proliferation actually increases by virtue of our physical movement within the environment. It's also influenced negatively in a variety of different diseases that influence the rate of proliferation as well as survival. And it's an area of great interest, and certainly the folks here at Erlangen have pursued this and demonstrated repeatedly its role in a variety of diseases. The final this continues to be investigated. One of my colleagues in Sweden recently used carbon dating, which was used for detecting how old fossils are, but they used carbon dating in the brain to determine whether or not, or to confirm whether or not new neurons were born late in life or embryonically. And they could confirm, using this advanced technology, that new neurons are being born in the human brain well into the 6th decade, which I was very happy to hear about, I must say. So the question is, what are these cells doing? And as I mentioned, these new cells, the adult-born neurons, are important for detecting small differences between closely related objects or events that occur. So here's an experiment that was done by Craig Stark, a very talented neurobiologist. And what he did was to take patients and put them into an fMRI machine and presented them with events, objects. And there's two parts of the experiment. There was an encoding experiment where they observed these items flashing at them over time. And then there was the test phase. And during the test phase, they would present either the same object as a repetition, a completely new object, or what they call a foil, which was an object with something like the one you'd seen before, but slightly different. And it wasn't so much asking the individual whether or not they could recognize this or not, but rather how their brain responded to these changes. Because remember, they're in a functional MRI machine and they're measuring blood glucose movement. So here's what the results would be. Here's the repeat. This is a repeat situation where the individual in the machine sees the flower. A repeat occurs, and this indicates that there is a large response in both sides of the dentate gyrus. Now, when you repeat it that second time here, that's the first response, and now the second response elicits a very small activation, and that's because the brain recognizes that it's seen it before. So it's a clear recognition, and the structure of the dentate gyrus doesn't respond. Now, in the case of a lure, you show the picture of a duck, and then here's the slight change in the duck, and you can detect it because they're sitting next to each other. And here's what the dentate gyrus now fires up. So if it had been exactly the same, it would not respond. But this particular individual's brain is using these new neurons in order to visualize or make this distinction between them. So here comes the test. Now, I want you to study these pictures very carefully, submit them to your hippocampus, and then at a later time point, I'm going to present these figures to you in conjunction with another figure. And all you have to do is to determine which one you've seen before of the two that I'm going to present. So you can either raise your hand if you want to, or just recognize what I'm saying. So what you want to do is indicate which one is the one that you've seen before by raising your hand or not. I don't have to do this too much. So most of you can nod if you think you understand. You figure you can think you know where it is. Okay? Do we get a lot of recognition out there? You can even raise your hand if you think you know which one it is. How about this? Yep. This is a very smart group here. How about this one? Okay, now you've got it. Okay, so this is this. Okay, so maybe this one. Okay, now I just want to tell you, your dentate gyrus is firing up at this point. So these are the cases when objects are closely related to each other. In those other cases, you didn't need your dentate gyrus, you didn't need any new objects. You could have solved this on other ways. But in situations where you are having to draw up the memory that you had before and make that comparison, that's where it's important. So I'll end this portion by just saying that we do study adult neurogenesis both from a molecular perspective, from an anatomical perspective, and from a disease perspective as the most remarkable aspect of brain plasticity that exists in the human body. So the brain is the organ that controls our behavior, and neural stem cells continue to make more neurons and connections throughout life. The ability to make new neurons is dependent on your behavior. So what you do actually can influence your brain structure and how well you can remember things. Your behavior thus affects the structure of the brain, which in turn controls your behavior. So you are who you are by virtue of what you do to a real anatomical extent. Thus, learning a musical instrument or gaining new scientific insights can generate and activate new neurons, which help you to appreciate the finer details of life. So in the last part, in this next part, I'm going to tell you about some new data that I'm doing. It's unrelated to this, and actually branching out into an area that I've become more interested in over the last 5 or so years. And this is in mental health issues, in particular in areas of psychiatric disorders. And I'll tell you a new story and some new findings that we have. And this focuses on a disease that's called bipolar disease or manic depression. It's characterized by unpredictable swings in mood from mania to depression, and it affects proportionally men and women, but women seem to be more affected by depression and not as many manic episodes. There are many different forms of manic depression, but in this study I focused on type 1, which are individuals that phase through this very high manic phase. It's a severe disease, at least in the United States, but it is true worldwide. It's estimated that 3 to 4% of the population are bipolar or have bipolar episodes. The bipolar disorder results in 9 to years of reduction of lifespan, so they age more quickly. There's evidence that there's both schizophrenia and bipolar age much more quickly. We don't understand this process, but they do. 1 in 5 patients with bipolar commit suicide, and 70% of the bipolars are misdiagnosed for years before they're finally treated effectively. Now, there are treatments for bipolar. That the primary treatment is something called lithium. It's an ion that, for which we really don't understand its mechanism, but anticonvulsants and antipsychotics are also used. The problem is that more than 50% of the patients that are diagnosed with this disease are what's called treatment resistant. And while if they are treated, it's treated in such a significant way that they can continue on their life and live quite effective lives. And the sad part of many of these diseases is that the underlying biological mechanism is essentially unknown. Recent advances that Jürgen alluded to using stem cells and what we call induced pluripotency has given us the opportunity to look inside the brains, or at least look at the brain cells of these patients. Just in summary, it was a technique that was sought after for about 10 years by many people, and included our lab. But in 2007, a Japanese scientist, Shinya Yamanaka, and his student Takahashi found the magic for transcription factors that could reverse a somatic cell biologically into an embryonic stem cell. It's almost magical, but it happens. People use this all the day, and it's being done in your university as well. So you can take a patient with the disease, and by molecular engineering, you can reverse that somatic cell from the body into an embryonic stem cell, which then can be induced into neurons in the brain. You can observe that patient's neurons in the brain. And what's pretty amazing is that they reflect biologically the activity of those cells. A key to this is then what type of cells do you differentiate them into, and I'll tell you a little story about that. But the idea is that once we search for that, look for molecular and physiological cues, the patient's own cells can be used to screen for new drugs, or even to use diagnostically to understand more about that individual's disease. A form of personalized medicine is on the break. What we know pathologically, pathobiologically about this disease is very little, but there is clearly this area of the hippocampus is shrunken, and the loss of, or the shrinkage of these cells in the dentate gyrus has been very important. So we've focused our attention on trying to make authentic dentate granule cells from patients with the disease so we could access very uniquely their cells. Just briefly, as always in science, we stand on the shoulders of giants, but there's about 40 years of research on developmental neurobiology, where we've understood specific molecules that are important for making the hippocampus, in particular dentate gyrus. We've taken advantage of that, and other markers that identify these cells uniquely. So here's a gene called PROX1 that's only expressed in the dentate gyrus, and here's in mouse, but it's also true of other species. We use these tools to identify methodologies that allow us to take the most primitive cell of an embryonic stem cell and make an authentic granule neuron. And we've developed protocols and disseminated those among our colleagues, and they work very effectively, showing all the hallmarks of these cells. And we can make these specific dentate granule neurons in vitro, and they behave electrophysiologically, spiking with appropriate frequency, as would happen in the central nervous system. We further demonstrated the authenticity of these by transplanting the human cells into an immunocompromised mouse. That means they have a human immune system, and we look for their ability to integrate within the dentate gyrus. These human cells not only become granule neurons within the dentate, but they send their processes out and make contact with targeted cells. We can take these cells and we can record from them with electrodes and show that these cells are electrophysiologically active in vivo, but also that they are receiving inputs and can evoke what's called a long-term potentiated response that is blocked by the appropriate pharmacological blockers. Now, some years ago, we published a paper in schizophrenia, a similar type of study looking at schizophrenia, where we used not this protocol, but a more general protocol for neurons, and we searched for several years for physiological differences between the two, but the variability was so high. And this is because the cells in the early days when we were differentiating them were not of a specific type, but of a more general type. So these were all of our early attempts to find differences. We took those same cells now using our current protocol, and we're able very clearly to see that schizophrenics' brain cells have a reduced synaptic connectivity that is reflected and very quantifiable. So armed with this ability to generate cells into a specific cell type and to detect differences in the function of these cells, we moved to bipolar disease. And I was encouraged to do this by many of my psychiatry friends who convinced me that this was really an unmet area of investigation. But we thought about how we could approach this in the smartest way possible that would allow us to take a feature of the disease that we could potentially model in vitro. And one of the things that I mentioned to you already is that there are patients within this population that respond very well to lithium and can live a quite normal life. And there's another population that are indistinguishable in terms of their phenotypic display to the psychiatrist that are completely unresponsive. So as part of a large consortium in bipolar, international consortium of bipolar, we're selecting patients that fall on the extremes of these two, responsive, non-responsive, and we brought these cells into our laboratory, reprogrammed them back into iPS cells, and this is just to give the unaficionados this conversion where they actually become quite reliably embryonic stem cells, but we can also differentiate them into neurons. And importantly, there's no real difference between the bipolar and the controls with regards to their capacity to be differentiated or reprogrammed. So that holds quite well. But when we took the cells that had been differentiated into neurons and we did what's called an RNA sequencing or whole genome RNA sequencing, the differences stood out dramatically. So we've really honed in here and looked at hippocampal neurons or dentate granule neurons that we've differentiated from the bipolar versus the controls. And it was really quite striking. There are orders of magnitude difference in some of these genes. And this clustering of these genes that many of you wouldn't be familiar with, but they're indicative of the idea that the cells may have an unstable hyperexcitability about them, these transcriptions. And we found hundreds of genes that were very highly significantly different from them. So that alone, I think, is really remarkable, that here you've taken a skin cell, converted it into a neuron, and done a whole RNA sequence, and you can detect differences between the patients, a large number of patients, and the controls. So we began the route of trying to physiologically see if we could find differences between them. All the cells had firing patterns, but very quickly and quite reliably, this came up, and this was repeated several times by different physiologists in the lab. But by patch clamping them, that is, holding the electrophysiological signal, a normal firing pattern of our granule cells would fire periodically, but these patients with bipolar had a very rapid, high-amplitude firing that was persistent for a long period of time, particularly in the early stages of the differentiation process. We have lots of parameters that we explored with regards to the ion channels that are involved in making this. We learned something quite functional about this as well, but it was a very reliable event. So one of the questions we wanted to do was that, is this pharmacological response, this lithium response, is it carried over to their cells? Can we actually see a difference? And this was you know, in science you have these great times sometimes where it does work out. This was a situation where the first thing we did was that we did the RNA-seq again after we gave the cells lithium for weeks in vitro, and then we analyzed what the response was to the lithium treatment. And in the responders, we had something like 400 genes that were statistically affected by lithium in vitro, but the non-responders didn't respond at all. The genes didn't react, so it's almost like they were completely different patients. They want me to continue to get online here. And with that gene data, we then went to the cells physiologically, and you can just take this as the evoked responses, the action potentials, where we see in the responders, we see that lithium could drop this hyperexcitability where the non-responders were unaffected. We've repeated this with a new cohort, and it's almost as if the non-responders actually respond worse to lithium than normals. And this is consistent on a lot of the parameters. But that's one cell at a time. We've done this differently where we've used a method called calcium imaging transients where we can monitor hundreds and thousands of cells simultaneously, so we get an index of the global activity and the connectivity, and we can demonstrate that this is a neural response. And if you look at the calcium events frequency, we see that there's a hyperexcitability certainly in the depressed group, and with lithium you can reduce this back to the normal level, but lithium again has no effect on these cells. Just as we were pushing our luck here, looking at other genes, families of genes that were not involved, and also trying to understand more of the mechanism. One thing that came up very clear was there was also not just a physiological hyperexcitability, but the mitochondria. These are the organelles within the cell that are the sources of energy, that make energy, ATP, for the function of the cells. They were seriously malfunctioning within the bipolar patients, and we documented this both functionally, morphologically, and what we did show is that the morphological features of the so what happens in the bipolar patients is the mitochondria, which are normally long, elongated, they fragment and they're degenerated, and many of you understand this process. But with chronic treatment in vitro, we could protect these mitochondria, and they became, in many ways, at least morphologically functional. However, when we looked at the function of the mitochondria in some detail, this hyperexcitability of the mitochondria that we saw in the bipolar patients was not affected. We didn't give up at this point because it may be that this mitochondrial defect is the initiating event that is fundamental, and so this is where we're spending a lot of our time. But we did wanted to see whether or not we could get begin to screen these cells for other drugs other than lithium that might be effective. Lamotrigine is a drug that's primarily used for epilepsy, and maybe some of you can see the similarity between what we're seeing here was this hyperexcitability and the theory that maybe an anti-epileptic drug at low doses might be able to suppress the hyperexcitability like it does with seizures. And sure enough, you can see here the normal individual with a normal spike activity, evoked response. Here in the non-responders, but also the responders, show this prolonged hyper-responsiveness to a single burst, which could be, in treating with lamotrigine, could block that tail. And now we know that our colleagues, both in the bipolar community, are working more at looking at different types of anticonvulsants to be used for treating. One of the things that's also happening now is we're finding ways to more quickly take the biopsy, screen the cells to see, as a patient's diagnosed, whether or not they're going to be lithium responders or not, because it often takes up to 3 years to really diagnose them appropriately for the response. So we're thinking that we're hopeful in our colleagues as well that there may be some diagnostic work as well as some therapeutic work. So in summary, Bipolar patients, where we derive these hippocampal cells from their own biopsies, we can detect a reliable hyperexcitability as a diagnostic for the disease. And the ability of lithium to reverse this in the patients is reflected in their cells as well, allowing for a model now to understand the mechanism of how this drug does work in some patients and not work in others. The mitochondrial deficit is not reversed by lithium, and this is room for us now to look for things that we can reverse or ameliorate to some extent that mitochondrial. And this is a little bit of speculation in my last slide here. To say that in this modeling, cell type specific or getting the right cell to model is crucial, we believe. Another feature that I comes up with my clinical friends is demonstrating that within a dish you can model a disease like bipolar or schizophrenia or autism, reveals that these are not deficits or diseases that are caused by bad parenting. These are biological diseases, just like Parkinson's, just like Alzheimer's disease, and should be treated as diseases and not as social diseases. I think I'll leave it there because I did mention that we're moving on with personalized medicine. I make a brief acknowledgement to my clinical colleagues who continue to be very encouraging, wanting me to focus on this and nothing else, to my chagrin. And of course, my sincere colleagues that have worked in this study. And I want to thank you for your attention, and thank you for honoring me with the Jakob-Herz-Preis. Thank you.